All right, today we're going to dive into the absolutely incredible story of penicillin. Seriously, it's one of the most important discoveries ever, and it's a wild tale of accidents, pure genius, and yeah, a little bit of mold that changed everything.
So let me start by asking you a question that probably sounds a little weird to us today. Imagine you're just playing outside, you get a little cut, or maybe you get a scratch from a thorn on a rose bush. Could something that small, that simple, actually kill you?
I know it's hard to wrap your head around, but for pretty much all of human history, the answer was a terrifying yes. A common bacteria could get into a tiny wound and turn it into a deadly infection. And doctors, well, they didn't really have a reliable way to stop it. This really sets the scene for a world that was very, very different from the one we live in.
To really get why penicillin was such a massive deal, we have to go back in time, and our story really kicks off on the battlefields of World War I with a young doctor from Scotland named Alexander Fleming. He saw a huge, terrible problem up close.
Fleming saw that so many soldiers weren't actually dying from bullets or explosions. They were dying from the infections that happened after they were wounded. And the crazy thing was, the treatments they were using, called antiseptics, were supposed to help. But Fleming started to realize they were often doing more harm than good.
So what even is an antiseptic? Well, think of it like a really powerful chemical cleaner. It's awesome at killing germs on a countertop, but when you pour it into a deep wound, it's a total disaster. Fleming actually proved that these harsh chemicals were destroying the soldiers' white blood cells. You know, the very cells that are our body's own personal army for fighting off infections.
So after the war, Fleming goes back to his lab in London, and he's totally obsessed with this idea. There has to be a way to kill bacteria without hurting the patient. And this right here is where the story takes a legendary turn, all thanks to a little bit of luck and a very sharp eye.
Now, Fleming was known for being a brilliant scientist, but let's just say he wasn't the tidiest person. So in the summer of 1928, he heads off on vacation, and he leaves a whole stack of petri dishes where he was growing bacteria just sitting out on his lab bench.
When he gets back, he starts to clean up the mess, but he stops at one specific dish. He notices a patch of blue-green mold has grown on it, and all around that mold, in a perfect little circle, the dangerous bacteria had been totally wiped out. Now, most people would have just tossed it in the trash, but Fleming saw something amazing was happening. He looked at it and reportedly just said, "That's funny."
Right away, Fleming realized he was looking at something completely new. This mold juice, as he called it, was making the substance that could kill germs but was harmless to human cells. He had just discovered the world's first antibiotic. If the antiseptic was a sledgehammer, this thing was like a smart missile that only took out the enemy. He named it penicillin.
Now you'd think, okay, great. Story over, he made a huge discovery, right? Well, not so fast. Here's the twist. Having a breakthrough is one thing, being able to actually use it is a whole other story. And this is where Fleming hit a giant wall.
He ran into three massive problems. First, growing the mold was super hard. You couldn't get very much of it. Second, the active ingredient in the mold juice was incredibly unstable. It would lose its germ-killing power almost instantly. And third, Fleming was a bacteriologist, an expert on bacteria, not a chemist. He just didn't have the right skills to purify the stuff. So he published what he found in 1929. But for almost 10 years, nothing. The discovery just sat there, a cool idea that nobody could use.
The story honestly could have just ended right there. But thankfully, nearly a decade later, with another world war about to start, a team of scientists at Oxford University just happened to find Fleming's old research paper. They saw the incredible potential and decided, okay, we're going to take on the challenge that Fleming couldn't solve. And this team, led by Howard Florey and Ernst Chain, was perfect for the job. Chain was a brilliant chemist, exactly the skill they needed. They worked like crazy, growing the mold in literally anything they could get their hands on. Bedpans, food tins, even old bathtubs. They had one single mission: turn that mold juice into a pure, stable drug that could actually save lives.
In 1940, it was time for the moment of truth. They took 8 mice and gave all of them a lethal dose of bacteria. 4 of the mice, the control group, were left alone. The other 4 got shots of purified penicillin. The result was just stunning. It couldn't have been clearer. The untreated mice all died. The mice that got penicillin, every single one of them survived.
So they tried it on their first human patient, a policeman who had a terrible infection from a simple scratch.
At first, it was like a miracle. He started getting better almost immediately, but their supply was so tiny that they ran out after just a few days. And tragically, the infection came back and he died. This showed them two things. One, the drug absolutely worked. And two, they had a massive new problem. They needed to figure out how to make a lot more of it.
With Britain in the middle of World War II, making the drug on a giant scale was just impossible there. So Florey took a huge gamble. He flew across the Atlantic to the United States to ask for help.
And in the US, the whole project just exploded. Scientists figured out that if you fed the mold something called corn steep liquor, which is basically a waste product for making cornstarch, it grew like crazy. Then, get this, after a nationwide search for better mold, they found a super potent version growing on a moldy cantaloupe from a local market. By 1944, factories were using these tricks to brew penicillin in gigantic tanks, making enough to treat every Allied soldier for the D-Day invasion.
Penicillin quickly earned its nickname, the Wonder Drug. It saved millions of lives during the war, treating infections from wounds that just a few years earlier would have been an almost certain death sentence.
For all their incredible work, Alexander Fleming, Howard Florey, and Ernst Chain all shared the Nobel Prize in 1945. But the real prize, for all of us, was the brand new era of medicine that they kicked off.
You see, this was so much bigger than just one medicine. Penicillin started the age of antibiotics. All of a sudden, deadly infections became simple to treat. Complex surgeries, from C-sections to open-heart surgery, became way safer. And because of that, life expectancy shot up all around the world.
So this discovery completely changed our relationship with disease. But it also created a new problem, which brings us to the final and maybe most important part of this story.
Turns out, Fleming himself actually warned everyone about this. In his Nobel Prize speech, he said he was worried that if we use penicillin too much or in the wrong way, the bacteria would eventually evolve and learn how to fight back. And you know what? He was exactly right. Today, antibiotic resistance is one of the biggest threats to our health, and it's making some infections really hard or sometimes even impossible to treat again.
The story of penicillin is just amazing. It shows us that a single aha moment in a messy lab is really just the beginning. It took a decade of waiting, a world war, brilliant teamwork, and even a moldy piece of fruit to turn that discovery into a true miracle. It's a huge reminder that science takes persistence and collaboration. And it leaves us with a really important question. What's our responsibility to use these incredible medicines wisely so they're still around to save lives for generations to come?